We'll go ahead and get started. So our first talk this morning is going to be on the microbiome in PSC, Hope or Hype, and this is going to be presented by James Tabibian. James is the Director of Endoscopy and Resident Research Director, Department of Medicine at Olive View UCLA Medical Center, and a Clinical Associate Professor at the UCLA School of Medicine. He's also a member of the PSC Scientific and Medical Advisory Board, and we were fortunate enough actually to have recruited James to Sacramento, but unfortunately Los Angeles was more attractive than Sacramento. Can't imagine why. If you go outside, you'll realize it's already like 90 degrees. So anyway, it's a real pleasure to have James back up here in Sacramento and to present on this topic of the microbiome. Let's see if I can get started.
All right, good morning, everyone. So after last night's fashion show up here, I was inspired and put in some crazy content into my presentation. And then this morning I woke up and said, maybe I'll be a little bit more grounded and just kind of focus on the facts as much as possible, less fashion or sensationalism and more just kind of science if possible. So I have no relevant financial disclosures. I should say I'm an advanced endoscopist, which is, I guess, good and all. However, on the scale of smarts, advanced endoscopists are not always the brightest people out there. So take that with a grain of salt. The hepatologists, on the other hand, are much heavier when it comes to smarts and perspective. So our objectives with this presentation are to provide an overview of the microbiome and the gut-liver axis, as it's called, as relevant to PSC. We'll also review some of the basic translational and human data supporting their importance in the pathogenesis and also potential treatment of PSC. And then hopefully we'll comment on some future directions and areas that might be meriting further study. Here's a diagram of, let's see if the mouse shows up, it does show up, of of the gut-liver axis, if you will. This is the small intestine here, or the intestines in general. This is the microbiome, which consists of bacteria, fungi, viruses, and all kinds of different metabolites. 70% of the blood that the liver receives, which I'll mention in a moment, actually comes from this outflow. This is the portal vein, eventually, that leads into the liver, and we refer to this as the enterohepatic circulation, which is, I think, in my mind and that of many, at the center of the gut-liver axis. So gut-liver interactions, as all of you know, are quite complex. It's an intricate interplay with many interrelated and moving parts and inputs. So into this interplay go host genes, both the genome as well as the epigenome, which is a whole separate topic in and of itself. The environment, including xenobiotics, diet, antibiotics, and also the enteric microbiome, which includes not only the microbiota itself, so the bacteria, the fungi, the viruses, but also their metabolites and also their degradative products. So the microbiome and the gut-liver axis are increasingly recognized as important in the maintenance of liver health and homeostasis, and it seems that perturbations in the microbiome might lead to the initiation of and or impact the features and progression of selected liver diseases, in particular PSC. So over the last few weeks, I thought to myself, how many PubMed publications are there about PSC and the microbiome? And PubMed, as you might know, is a collection of the National Library of Medicine's scientific works. And I searched primary sclerosing cholangitis. I got over 6,000 hits, which is great, going all the way back to, I believe, the 1950s. I then searched cholangitis sclerosing, and MESH also doesn't have its own term for primary sclerosing cholangitis, but hopefully with the advent of an ICD-10 code, that might change. So I searched that together with gastrointestinal microbiome. I got 20 hits ever published. So that can't be real. There's more than that. I think Dr. Bolus has probably written 30 himself. So then I searched a slightly different technique. I said cholangitis sclerosing and microbiota. I got 30 hits. I said, okay, there might be more on the way. So I searched a different way. I just typed in microbiome primary sclerosing cholangitis. I got 66 hits. And I think there's actually more out there. But interestingly, it all started in 2011, at least if you search with any one of those criteria. So prior to then, there really wasn't that much chatter or scientific dialogue about the microbiome in PSC. So I think my first take-home message, I'll have a few of these peppered in along the way, is that the microbiome is mesmerizingly complex, and it's a prominent topic in PSC circles, especially in recent years. The microbiome research in PSC is still really a small sliver of microbiome research in general, which if you type in microbiome, you get over 43,000 hits. And even in the context of liver disease, it's not that big on the radar. In fact, I came across a very nice review about the microbiota in the liver, published in one of the official journals of the U.S. Liver Society, AASLD, and the word primary sclerosing cholangitis, or PSC, doesn't show up. I was very surprised. That being said, it is gaining increasing traction and attention as an area with great potential in PSC. So what supports this notion of a gut-liver axis in PSC? And as I mentioned earlier, 70% of the liver's blood supply typically comes from the outflow of the intestines. Intestinal metabolites, bacteria, and other substances and molecules enter the liver through the portal vein, which is the liver's major blood supply. The bile duct cells, or cholangiocytes, or biliary epithelial cells, they all kind of imply the same thing, in PSC seem to accumulate lipopolysaccharide, or LPS, which is a structural molecule in the cell wall of bacteria. And PSC cholangiocytes, and also to some extent PBC cholangiocytes, seem to be hypersensitive to LPS. In other words, if you take them in a petri dish and you take these cholangiocytes and you introduce LPS into the dish, they seem to react to it much more prominently than normal cholangiocytes or normal bile duct cells. You all know about the PSC-IBD relationship, which in and of itself is an amazing association. So that itself, to me and to many, says that there is some kind of link to the gut, to the microbiome. There are also associations with genomic loci, or genes, that are involved in host microbiome interactions. So I've just mentioned or listed a couple here, but there are others as well that stand out in PSC and suggest that one thing that makes patients with PSC unique is that they might have some genetic differences or changes in genes that affect how the body interacts with microbes. There's also intriguing model system, in vitro model, animal model data, as well as clinical trial data that pertain to the microbiome and antibiotics, and I'll come back to those. And also alterations in the microbiome that seem to be unique to patients with PSC. So let's talk about that for a moment. So patients with PSC have a marked decrease in intestinal microbial diversity. In addition, there's also intestinal dysbiosis, in other words, an imbalance in the bacteria and also therefore their metabolites and other molecules. So on the one hand, there's overrepresentation of numerous genera or genuses of bacteria compared to healthy individuals, as well as individuals who just have IBD but no PSC. And at the same time, there's also reductions in various other genera, also compared to healthy controls and patients with IBD only. So not only is the diversity of overall microbes decreased in the gut microbiome of patients with PSC, but the balance is also somewhat off, and with a very unique signature, I'll say as well. So the second take-home message is that there are numerous observations and lines of evidence that support the notion that there's an important association between the microbiome and PSC, and this association might impact who develops PSC, what their PSC looks like. Is there also IBD, no IBD? Is it small duct only, large duct only PSC, and also how it will progress? And these are areas of active and really exciting research, I think. So I'm going to branch off now and come back to what I mentioned a few slides ago and provide some selected examples that I think demonstrate the association between the microbiome and the PSC. And I'm going to share with you a few slides about from animal models, as well as translational studies, which are we use that term to, in general, suggest a bridge, if you will, between basic science and also clinical or human studies. And then also, I'll share some data from clinical trials, which I think show some promising experiences with oral antibiotics. I do want to emphasize that given the time constraints, this is not going to be an exhaustive list of interesting data, and there are numerous other studies that support this important notion of the association between the microbiome and PSC. So many of you have probably heard of something called the MDR2 knockout mouse. It's a mouse model that's typically used or often used to study sclerosing cholangitis, not necessarily only primary sclerosing cholangitis, but sclerosing cholangitis in general. The way that the model works is that it's missing a certain gene that, as a result of that missing gene, bile tends to be more injurious or hydrophobic, and it irritates the bile ducts. And here you can see a wild type or normal mouse biliary system. This is taken out of the mouse and injected with a polymer to make it kind of red and stand out. Here's an MDR2 knockout mouse bile duct system, which you can tell is very ragged. It has some dilations, has probably some narrowings. And this is an MRI of a live mouse that really kind of shows or recapitulates these changes, which are very reminiscent of PSC. Now, that's great, but until very recently, there have been no data regarding the role or the impact of the microbiome in this mouse model. So we asked a few years ago, does the absence of the microbiome influence disease phenotype in this model of PSC? So we had two groups of mice, conventionally housed mice, or CH, as you can see here, and also germ-free mice, MDR2 knockout. Both were MDR2 knockout. These are abbreviated with GF. And we know that this phenotype, the conventionally housed mice, in other words, they have bacteria, they have the whole, fungi, viruses, everything that would be normal to a mouse. These mice develop sclerosing cholangitis, whereas these, we really don't know what happens. And so these were mice that were matched on age and sex and strain, and we sacrificed them at about 60 days to see what does their disease look like, comparing these germ-free mice to the conventionally housed. And what we essentially found was that the germ-free mice demonstrate more severe serum biochemical abnormalities, and that was really present across the board. So the typical markers that we measure in humans, alkaline phosphatase, AST, bilirubin, all of those were much more abnormal in the germ-free mice compared to the conventionally housed. The same can be said from a histological perspective. So when we examined the livers of these mice, we saw that the germ-free mice exhibit more scarring or more fibrosis, more ductular reaction, which is a sign of injury, and also ductopenia, so loss of bile ducts compared to the conventionally housed mice. And here you can see in this column, this is fibrosis or scarring. The germ-free mice are on the bottom row. There's a lot more of this bluish scarring compared to the conventionally housed mice. There's much more ductular reaction in this column. A lot of cells here that are poorly organized. They're sort of trying to form new bile ducts, but are not successful. And then here, ductopenia. This is the MDR2. This is the conventionally housed mice, which has some periductal onion skinning here. This is a bile duct. Whereas in the germ-free mouse, you kind of lose the bile duct, and instead it's replaced with poorly organized ductular reaction. So based on really that first germ-free variant of the MDR2 knockout mouse model, I think it's safe to say that the germ-free state has unambiguous effects on the features of disease in the mouse. And specifically, there's serologic evidence of more liver inflammation and cholestasis, or bile not flowing, and also increased liver fibrosis, ductular reaction, and ductopenia. To follow up on that study, my mentor, Dr. LaRusso, and colleagues tried to see if we kind of go the other route. We take a conventionally housed mice and then essentially blast it with antibiotics to wipe out its normal gut bacteria. What happens? Do we see a similar picture? Is the disease worse when the normal microbiome was lacking? And these are unpublished data, but you can see essentially in the blue bars that the mice treated with broad-spectrum antibiotics, again, biochemically seem to have more severe disease. The same can be said about fibrosis in the liver. The mice treated with antibiotics have more scarring, and therefore it seems like they do worse. So take-home message 3 is that the absence of the normal intestinal microbiome makes a mouse model of PSC have significantly more severe disease. Moving on to a translational study we did not long ago, looking at the enterohepatic metabolome, or the metabolome of the enterohepatic circulation in PSC. And our goal was to quantitatively and qualitatively examine and compare, using metabolomics techniques, profiles in portal vein blood, peripheral blood, and also bile in 3 groups of patients: patients with PSC, healthy controls, in other words, living donors. These are folks who donated a portion or a lobe of their liver, and also patients who are undergoing liver transplant for reasons other than PSC, like PBC, autoimmune hepatitis, hepatitis C, fatty liver. And I'm going to fly through these slides because some of the data analysis is not yet complete. This is shared with me again by Dr. LaRusso and Steve O'Hara. But what has been found, or what we found, was that the enterohepatic and serum metabolomic profiles in PSC differ from healthy as well as disease controls. And what you want to be looking at here is the clustering or the separation of the different groups. So the PSC patients, shown in blue, seem to kind of stand alone in each of these 3 diagrams. There is some overlap compared to healthy controls in green and disease controls in red. So this clustering suggests that there is a difference, and it was most pronounced actually in portal serum or portal blood and peripheral blood. So take-home message 4 is that there's a clear biochemical signal that the enterohepatic and serum metabolomic profiles are unique in PSC, and that they're different from healthy as well as other disease controls. Clinical trial data. So, as many of you know, the human experience thus far supports the notion of the importance of the microbiome in PSC, and really it's quite exciting, the findings thus far. A few years ago with Dr. Lindor, who's a friend of the PSC community, as many of you know, Dr. Lindor and I worked on a study that was ultimately published entitled Randomized Clinical Trial, Vancomycin or Metronidazole in Patients with PSC, a pilot study. So the aim of this study was to prospectively investigate the safety and efficacy of oral vancomycin and oral metronidazole in patients with PSC. And this was a 12-week or 3-month study, single center, and we randomized patients with PSC to 4 groups: low-dose vancomycin, high-dose vancomycin, low-dose metronidazole, and high-dose metronidazole. And the inclusion criteria were that patients had to have an elevated alkaline phosphatase for at least 6 months prior to the study, and patients with cirrhosis were not included, so only patients with earlier stage disease. 35 patients were enrolled and assigned to the 4 treatment arms. The median age was 40 years, 60% were male, and 71% had IBD, which is pretty representative of IBD in general. And the median baseline alkaline phosphatase value at study entry was 383, so it was above the at least 1.5 times the upper limit of normal in the laboratory. And there were no significant differences between the 4 arms, except for the vancomycin arm seemed to have lower fatigue. This is grossly simplifying the study findings, but as many of you know, what we found was that low-dose vancomycin and even high-dose vancomycin seemed to show a significant reduction in alkaline phosphatase, about a 45% reduction in both arms, whereas metronidazole didn't really seem to have this relationship with alkaline phosphatase reduction. So I think take-home message 5 is that vancomycin and even metronidazole, which I'll show you in a second, seems to show promise in treating patients with PSC. So summarizing the clinical trial endpoints, the primary endpoint was alkaline phosphatase. Secondary endpoints were several, and they were Mayo risk score, total bilirubin, CRP, or C-reactive protein, and pruritus itching. And although only the vancomycin groups showed a response in the primary endpoint of alkaline phosphatase reduction, the secondary endpoints actually, some of them were met in the metronidazole group. So to me, that suggests that even metronidazole might have some therapeutic promise. And anecdotally, I have seen that in patients with PSC who take metronidazole for perhaps an unrelated reason, and they say, hey, no, doc, in 7 days my itching went away. And so there's something to be said there, I think. We're wrapping up here slowly. So I think putting all of that together, I tried to develop together with mentors, a conceptual model of the microbiome and the gut-liver axis in PSC. So I don't think my animation is working, but essentially this is the diagram that I showed you earlier this morning with the intestine, the portal vein, the liver, and the thought is that some form of insult, probably or possibly microbial, tends to elicit an acute cholangiocyte or bile duct cell response. The enteric microbiome can either exacerbate that injury or the attempt to resolve the injury, or it can attenuate it, might make it better. And that probably depends on a number of things, but one of them is the composition of the microbiome. So on the one hand, a healthy microbiome might help put this injury to rest, and you might have another arrow here that goes to resolution of injury, or a diseased microbiome might lead to persistent insult or persistent host response, immune response, leading to chronic biliary injury and ultimately leading to development of PSC. And I think this arrow here could also go to the right and point up this way, because I do think that an abnormal microbiome or a dysbiotic microbiome could potentially make the course of PSC worse. So not only can it impact whether or not you develop PSC, but I think it might also impact how PSC does over time. So in conclusion, I do believe there's a growing basic translational and clinical body of data supporting the notion that the microbiome is vitally important in PSC. The dynamics of this relationship, as I've conveyed, are very complex, very intricate, and as much as I'd like to be very excited, which I am, even though I may not sound like it, I do need to be honest and real with myself and the scientific community and my patients and say that I think knowledge is in its early stages, but early doesn't mean it's bad, or early doesn't mean it's false or weak. It just means we still have work to do. And with that said, I think high throughput technologies, I'm using some fancy terms here, excuse me, gnotobiotic models, in other words, mouse models or other animal models where we know what are the organisms that we have placed in them so that we can selectively study a certain bacterium or a certain group of bacteria. Human microbiome studies off and on treatment, and also team science approaches I think are going to be paramount and are paramount for furthering our understanding and developing biomarkers and also treatments for PSC. So I think there certainly is some hype, but there's also a lot of hope to be had, and want to thank you all for your attention and thank the PSC partners seeking a cure for the opportunity to come and present. Thank you.